All right, thanks everybody for being here and getting up. I kept this relatively short, which, given everyone's hackathon hangovers, I think that's probably a good plan. So I'm going to talk a little bit about some of the reasons why we're interested in this topic, and then just going to go through some of the results that I've gotten so far using this kind of paradigm. And then for the last section, I'm going to go into a little more detail about exactly how the model works. Which I usually don't talk about in this when I talk on this topic, but I think you guys would be interested in. And I have lots of extra slides for all this stuff, so feel free to interrupt me at any point, and I can go on a very extended diatribe about whatever. Okay, so in Chris Honey's talk, and we've been hearing from a lot of people, there's really this key idea when we're trying to understand naturalistic stimuli in the world, and a quote that I like that sums this up. This is this book critic Pete Dexter is talking about why his favorite novel is this book, A River Runs Through It. And he says that except for the first sentence, in our family there was no clear line between religion and fly fishing. There isn't anything in it that doesn't depend on what comes before it for its meaning. Right? So this was his measure of sort of the best kind of story is one where every piece of information you get is sort of context dependent, and you really need to integrate it with things that you've already learned. And this general idea, I think, is present in almost any kind of complex understanding of narratives or of the real world. So for all the experiments I'm going to show you, we use relatively realistic videos and narratives. Most of these are from things like Hollywood movies and TV shows. So I'll just play one more. We've seen a lot of videos, so I'll play one more little video clip as an example for some of the things I'm going to discuss. So this is a relatively simple little scene, but if you think about how you actually process this, you really have to do a lot of keeping track of what's going on, right? To understand what they're doing on the floor there at the end, you really have to understand the kind of situation that they find themselves in, right? So definitely, intuitively, there's some idea we have to kind of keep track of situations over time. And again, a lot of the previous speakers have talked about this. So that both in these shows and in real life, often our situations don't actually change smoothly over time, but they're really divided into these events, right? So in this case, for example, right, there's sort of one situation when Cameron Diaz is going up the escalator, and there's a very sudden change in the kind of situation that's occurring, right? And so there's a change in our predictions about the future. There's a change in the kind of information we're keeping track of. And so these kind of rapid shifts in situations have been studied for decades in cognitive psychology and referred to as event boundaries. And so we have at least some evidence, most of this is from behavior, that these event boundaries are really critical for both perception and memory. So for example, if you just use eye tracking to time people that are reading a story, you find that people pause for about 1/3 of a second if they hit one of the situation boundaries in text. Implying that maybe there's some kind of specialized processing that's happening here. We know that these boundaries have an impact on sort of ongoing working memory. So for example, if when you cross one of these event boundaries, people find it harder to recall what had just happened in the movie on the other side of the event boundary. Right? This is also kind of like this phenomenon of walking into a room and forgetting why you went in. Right? Once you cross that event boundary of the room, you flush out a lot of your working memory, and it can make it hard to keep track of things. There's also some evidence that this event structure perception is actually important for the way that long-term memories get encoded. So if you do a cued association task, and that we find that people, this is work from Leila Tvashi's group, shows that people have a harder time associating stimuli that fall on opposite sides of an event boundary. Right? So the idea is that these event boundaries are important both to understand what's going on and also seem to structure the way that we actually remember these stories, right? So all of these, most of the recent experiments on event boundaries basically use things like lists of words and lists of pictures, or they have you change the task you're doing. And so there hasn't been much work recently on trying to look at these event boundaries in realistic narrative stimuli. And so one of the reasons people have not done this is because the events in narrative stimuli are a little bit harder to define, right? So if we define an event as one, face stimuli changing to scene stimuli, it's very obvious where the boundaries are. The experimenter controls them. The question is, in these kinds of narratives, what constitutes an event boundary? So one thing we could do is define event boundaries as whenever there's a big change in the low-level properties of the stimulus, which would give us things like camera angle changes in a movie. And this is an event in some sense, and I'll come back to this, but this is not the kind of events that people in cognitive psychology are thinking about. Right? Those events are things where the situation is changing, even if there's not a big change in the underlying stimulus. Okay, so we wanted to find things with situation changes, but there's another question here too about timescales, right? And so how big of a chunk of a situation should an event be? So right, in this clip I showed you, we have sort of two situations here, right? So maybe if we sort of keep track of like the last 10 seconds, that should be enough. But actually, I didn't show you the first half of this clip. So I'm going to play this part of the clip that I didn't show you. Right, so with some more context, right, we now see the situation is different than what we thought before, right? And so now there's sort of less like exploding exponential universe of possibilities here for what we should consider situations, right? Are these like shorter segments where a person is performing an action? Are they these like super long things where right, there's something that happens that then colors the context of many minutes in the future? So how do we start figuring out right, which of these the brain cares about, or which ones of these the brain cares about? So one approach is to basically just get a bunch of people and show them these kinds of stimuli and ask them where they think the event boundaries are. And you could ask them to do this at multiple temporal granularities, for example, right? Say, give me coarse, sort of minutes-long events, and then give me some finer ones. And then we could look to see when a person's watching this movie, in some particular brain area, do we see some response that's locked to these boundaries? Right, so we ask people where we think these boundaries are, and then we look in our fMRI data at these human-labeled boundaries and ask, is there something going on at the brain at these boundaries? And so you can get at least a little bit of traction with this. There's some brain areas that do show an increase in activity at these human-labeled boundaries. The problem is that if we really want to characterize what each region of the brain is doing, we're going to need a huge, huge number of these possible event boundaries, right? So we might have brain areas that are segmenting at different timescales. There could be brain areas that have the same timescale but care about different features, right? So maybe there's some brain areas that segment when you change location, and there's other brain areas that segment when you have a change in the social interaction. And so it starts getting a little bit impossible to actually try to get people to generate every possible hypothesis here. And so the idea is, can we run this analysis backwards? So given the fMRI data, can we predict where we think these event boundaries are occurring, right? So we're going to try to ask a brain region how we think, how it's segmenting up the world. And then if we want, we can later compare to human annotations or other things to try to understand what it means, right? But the idea here is we don't go in with specific hypotheses about where the events should be. We're trying to let the data tell us where they are. So there are a bunch of ways you could imagine trying to do this. So we picked what we thought was the simplest possible way to look at this, which is to say that when we have a region of the brain that rapidly changes its pattern of activity, that would indicate that this region thinks that a new event has started. So the idea is we're going to just get fMRI time courses. So there's no labeling here. We just get a voxel by time point matrix of fMRI data. And the idea is that we want to automatically detect when we're jumping between these stable patterns of activity. Right? So this is the kind of thing that we're building the model to try to detect when a brain region is stuck in some pattern of activity for a relatively long period of time, and then rapidly shifts to another pattern of activity. And so whether these shifts are actually event boundaries is something we'll have to try to show, right? But this is the one hypothesis for what event boundaries might look like in the brain. So we've been doing a bunch of analyses with this model, so I'm just going to sort of run through a lot of the different things we've tried so far. And so there's questions here that are solely about perception, and then there's questions about how this event structure influences memory. So let's start with the perception questions. So first of all, we can go back to this question of timescales, which has already shown up a few times, and ask what is the timescale of these events, and how does that change in different brain regions? So from Chris and Janice, we've been hearing about this timescale hierarchy, right? So here's a radically simplified figure of their results, right? Which is that in these early sensory regions, we should have relatively short timescales, meaning that they don't depend that much on prior context, whereas in these more default mode regions, we have these longer timescales. And there's also a parallel literature to this a little bit in the scene processing community and in the vision sciences community, which is where I was doing my PhD. So based on this work, we might guess that these short and long timescale areas correspond to short and long events, right? So these more sensory regions should divide up the stimuli more finely into events that are more based on sort of low-level changes in the stimulus, whereas these high-level regions should have patterns of activity that are relatively stable. So we can take the Sherlock data set, which you guys are now intimately familiar with, and so we're going to apply this model to this data set, and I'll talk at the end in a lot more detail about what that means. Right, but the idea is we're going to take data from some region of interest and try to figure out how many event boundaries there are and where they are. So here's just a little example. This is just showing a few minutes of this hour-long stimulus. So we can look in early visual cortex, for example, right? And every line here is where the model thinks there's a shift in activity. And so it has, hundreds or even more of patterns depending on how you define them. As we sort of proceed up the hierarchy, once we're in late visual cortex, we start seeing fewer events. And finally, up in angular gyrus, and in these posterior medial regions around posterior cingulate cortex have the slowest timescales. So there's a few things to notice. So first of all, the number of events is going down as we go up. And you can also see there's some alignment of these boundaries across regions. So it's certainly not perfect, but definitely highly above chance that if we have a boundary in these low-level regions, it also might appear in the higher-level ones, right? Or really to reverse that, the boundaries in the high-level regions, most of them have appeared in a lower-level region before, but we're also getting invariant to a lot of the shifts that weren't important to the situation. Yeah?
So how are you using the FMR data to identify the event contracts?
Yeah, so I'll talk in a lot more detail at the end about exactly how the model gets fit, right? It's basically just looking for when there's a shift in activity patterns, right? So so the idea here is that, right, so the fMRI, this voxel by time point matrix, right? The spatial pattern of activity is relatively constant, and then there's a shift, and now there's a new fMRI activity pattern here, right? So it's trying to learn this in an unsupervised way. When do these patterns shift? Yeah.
Cyclic of these event boundaries, do they appear like sort of within a set amount of time, or is that just there's no pattern, there's no segmentation which they?
Yeah. So I mean, certainly different regions, since the event length is different, right? So there's sort of an average time when the boundaries come up. But it's not super regular, right? So you can even see in the posterior medial one, right? There are these two events that are like really close to each other, right? And I think this is actually an important point about this, right? Which is a way that this method is a little bit different from things like autocorrelation methods, right? Or power spectral methods that basically assume that there should be a frequency at which these events are occurring. And so, I mean, here there is sort of a rough frequency, right? But it allows for the events to be much more uneven. Yeah.
So just to follow up on that, but on your side, I guess, if figuring out what is happening on a longer time scale helps me to have a predictive window of what might come next, as soon as you shift my schema or what is the thing, my predictive representations need to be updated. Assuming longer time scales care for predictive representations, it makes sense that they would shift immediately when there is an event that opens up a new predictive sort of schema, although they will stand longer. So
Right.
it's okay not to be on the frequent window.
Yeah, that's right. That's right, because it's locked to particular aspects of this particular stimulus, right? And so, yeah, so if this stimulus happens to have two quick situation shifts, you want these regions to change faster. Right, so yeah, so I think one important thing, right, to think about timescales in all these regions is those are just sort of roughly where they live in this timescale continuum, but it's really the properties of the stimulus that determine how they're doing the chunking. And so then, yeah, the last thing we can do is we can compare to where humans think these boundaries are. So, across different people, there's some disagreement among people about exactly how to divide up the movie. But there are on the order of about like 30 to 40 of these event boundaries that pretty much everyone agrees are an event boundary. And these match up best with these boundaries at the top of the hierarchy. Right, so as we're going from these lower level to these higher level regions, basically bunches of these boundaries are dropping out, and those are the boundaries that don't correspond to a meaningful change in the stimulus. All right, so this is just a little example I'll show you over a few minutes. So we can, again, look at these regions in the context of a whole searchlight over the whole brain. And this is what we get. So the regions in blue here are regions that have relatively fast events. So these are events where, these are regions where the model thinks there's lots of pattern shifts. And the regions in yellow are those where the model thinks there's very few pattern shifts. So there's relatively long periods of stable activity. And so this at least roughly lines up with Janice and Chris's timescale map. There's a supplementary figure in the paper where we try to quantitatively see how close they are, and at least the broad topography is similar. We do get early visual cortex as being short events, so that partially answers that mystery. I don't want to talk about that. And we can also see at least a little bit of this progression from short to long, right? So there's regions like up here on the angular gyrus, for example, that are certainly long-ish events, right? That are on the order of over 30 seconds or so, but are shorter than regions that are in posterior medial cortex, right? So we start to see a little bit of division even among these long regions, these long event regions, in exactly how long they are. Another little animation of this. So what I'm going to play here on the left is a sped up version of the fMRI data. So it's sped up about 30 times, so it's going to be kind of fast. And I'm highlighting here the angular gyrus. What I'm going to plot on the right is an MDS plot. So this is a kind of hypertool style plot of where we are in activity space in the angular gyrus. And so when the dot is close to this picture, that means that the angular gyrus is sort of looking like this. And when the dot is close to this picture, the angular gyrus is looking like this. And so the point here is to look at the temporal structure of this dot. And so I'll just start playing. Right, so the idea, this event hypothesis, right, is that basically the dot kind of goes to somewhere in activity space and lives there for 30 seconds to a minute or so, and then rapidly jumps to somewhere else in activity space. Right, and I'm coloring the dot here with which event we think that the dot is currently in. Right, so every time the dot jumps, then we're labeling it with a different color. Right, so you can see there is, certainly it's not that the dot jumps and then is 100% stationary and then jumps again, right? There's lots, it's like always doing lots of little wiggling. But there definitely are periods of greater stability when the pattern is relatively stable, and then sort of rapid jumps to a new part of activity space. Can you say what you mean? The transition probabilities. Yeah. So at least in the basic version of the model, every state is just independent, is just its own. Every time you go into a new event, it's just a new state that's unrelated to every other previous state, right? So you can post hoc look at like this event structure, right? So what events are similar to each other, and certainly there's a lot of structure there. So right, yeah, you'll revisit parts of activity space that are similar to before. Yeah. So you're saying about like if you're currently in a bottom left one, do you then go to the top left next or something? Yeah, we've been trying to look at that. One tool we've been using is these topological data analysis tools, which basically try to look for like little sequences of patterns or little loops of patterns and stuff. Yeah, it's a little tricky. I don't think I have anything super intelligent to say, but it's but that's certainly something I'm interested in. Yeah. Yeah, that's right. I mean, part of the problem with all of these things is that as soon as you start getting into these long events, then you have something like 50 events in your whole experiment, right? Because it's it's just hard to keep people in there too long. So, yeah, so I've been trying to think more about ways to, for example, if we showed people lots of different stimuli, where even the stimuli are different across different people, right? To try to get like a bigger sampling of these transitions. But yeah, I'm not sure the right way to do it.
That's really cool.
Yeah.
Do you see the same activity states across different stimuli? Or like really at least visually, even if you decompose them in seconds?
I mean, certainly there's activity states that are very correlated. So I could show the schema stuff at the end, right there. So we have another version of this where we have people watching a bunch of movies where we expect there to be similarity across the events, and so certainly there's some in that case. Yeah, it's a little hard to say. I mean, there's definitely similarity across the movies. You'd really have to show with an encoding model or something that it's like meaningful. But yeah, do you have a question? Okay.
Trying to understand whether you meant this to be within event structure or across event structure. So what are you considering an event for this visualization?
Yeah, so the events here are whenever the dot stays relatively stationary, right? So when it goes and stays in one portion of this space for for at least 10 to 20 seconds or something, right? That's an event, and then whenever it makes these big jumps, that's an event boundary, right? That the representation is changing quickly.
To follow up on Luke's comment, so you treat right now events as MVPs, but if you treat these states as MVPs, it's possible to get the sort of the probability transition matrix of them, even no matter how large, even if it's 50 by 50.
Right.
So
Yeah,
how
what's
do
Richard?
states and see whether there is a probability of transition for them that is specific to particular events? So this 9 states
Mm-hmm. Mm-hmm.
as the MVP.
Yeah.
Sequences or patterns or transition probability matrices as different events.
Right. Yeah, that's right. Yeah, so the 9 things here are just for visualization. So, but you're right. We could basically make these sort of bins that you're allowed to go back to, right? And then see how they transition. And so we've tried that a little bit. It's just like statistically a little hard because we only ever observe, right? Even if we have 9 bins, that's 81 transitions, and we only have 81 possible transitions, and we only ever see 50. So, we just don't have very good sampling of them. But okay, so so far we've basically both sort of replicated this previous work on timescales and also shown now that we can start identifying really where these little context chunks live in time. So then we can also ask what's being represented in these long timescale regions. And again, Janice has already basically answered this question about that. We think they're representing these relatively abstract situation models that represent sort of features of the situation, things like who's there, where is it, what time is it, what are people's goals, and is relatively abstracted away from the exact sensory details of how we learn that information. So Janice mentioned this data set of people. We have people watching Sherlock and then listening to an audio description of Sherlock. And so it's these 2 different data sets depict the same events, but in different modalities. So Janice already showed one way to analyze this data set is to just hand label which pieces of the movie and which pieces of the audio correspond to one another, and then see if their patterns look similar. So we can try to do something a little fancier here and don't have humans involved. We just have humans are involved as subjects, but so we have this movie data set, which is again sort of some number of voxels by some number of time points, and we have this audio data set, which is the same voxels but a different number of time points, right? And what we want to ask is whether these 2 data sets proceed through the same sequence of events. And so a way we can try to do that is by running this event segmentation model in the movie, right? So now we're going to describe this movie as this sequence of event patterns. And then we can ask the model, do we think you also see the same sequence of patterns in the audio data? Right? So the question is, can we basically align these 2 data sets by saying that they go through the same sequence of these latent event states, even though the amount of time you spend in each event might be different? And so the question is, can we do this automatic lining up? And the answer is that we can. And we can only do it in these relatively long timescale regions that we think represent this kind of situation models. Right, so this is relatively automated way of testing whether we think these data sets go through the same sequence of narrative steps. And we can just test if this model can actually fit in each region of the brain. And we find, again, in these sort of angular gyrus and posterior medial regions, as well as this inferior frontal gyrus, we get some lineup as well. All right, so then we can start to ask about how this event structure during perception influences long-term memory. So the first question is, how do we remember stuff in the real world? And so, right, the vast majority of memory research on episodic memory, for example, gives people basically a list of items, so pictures or words, and then has people try to remember them or remember associations between them. And so this is a great paradigm for studying lots of things, but it's not totally clear how this actually corresponds to something that you do in the real world, right? In the real world, you're not given a list of the things you're supposed to remember about the day. You just get this continuous stream of experience, and you have to do something with it. So one possible clue here comes from people with hippocampal amnesia. So this patient HM is the most famous version of this. Right, and so people with hippocampal amnesia are often very fluent, so they can have a conversation with you and they can do at least certain kinds of tasks, but they're unable to form new episodic memories, right? At least if they're having anterograde amnesia. And this type of memory deficit that they have is really specific to these kinds of event boundaries. Right, so you can have a conversation over the course of several minutes with someone that has hippocampal amnesia, and they'll be able to act normally. But if there's some type of contextual shift, right, so someone changes the topic of conversation, or you change the type of question that you're asking them, they then are unable to keep track of things across those boundaries. So there's obviously been a lot of interest in studying people that have this disorder. And the way that connects to the event boundaries, at least one possible hypothesis, is that these event boundaries critically depend on the hippocampus in some way for long-term memory, right? And so there's something that you need your hippocampus for to get across these event boundaries. And so one way that might happen is that there's some type of encoding process, some type of memory encoding process that happens specifically at these event boundaries that involves the hippocampus. Okay, so how are we going to look for this? So what we can do is take some region of the brain, apply our event segmentation model like we've been doing, right? And so this will give us time points in the movie where we think that there's an event shift. And then what we're going to do is look in the hippocampus, just in the univariate overall activity of the hippocampus at these event boundaries. And ask, is there something going on at the hippocampus at these event boundaries? Right, so what we do is we're going to define our event patterns, our event boundaries from the cortex. And then we're going to look to see if, on average, the hippocampus is doing something interesting at these event boundaries. So, for example, if we use angular gyrus as our cortical region, so right, we're going to define event boundaries based on when activity in angular gyrus changes quickly. We see that when angular gyrus changes quickly, we see this very large increase in hippocampal activity. This is a super robust effect. You can even get most of this if you just hand label the boundaries. There's a very big increase in hippocampal activity. We can also look across the brain what cortical regions have boundaries that are related to this hippocampal bump. And again, we find angular gyrus and posterior medial cortex, as well as some parahippocampal gyrus and a little bit of inferior temporal. Right, so this would argue for some kind of model where these regions are coupled to the hippocampus specifically at these event boundaries, right? That when this event boundary shifts, there's some kind of process happening in the hippocampus. Now, the data I've shown you so far shouldn't convince you that this is encoding specifically. I can go into this in a lot more detail if you guys are interested, but right, but at least there's like something interesting happening at the event boundaries in the hippocampus, and it could be at least some of it is related to the scene that's ending, and some of it might also be related to the scene that's starting. But now that we can identify these event boundaries automatically, we can start to ask what this sort of time-locked activity is. Yeah.
Can you go back to that?
Mm-hmm.
So would you expect that the increase would be 3 to 5 seconds delayed rather than
Right. So yeah, so both of these are hemodynamic signals, so we shouldn't expect any like hemodynamic lag between the two of them. But you're right that we should, if this is really about like something that happens at the end of the event, then it shouldn't start doing anything until the event's over, because how could it know the event is about to end before you're there, right? So I think there's a few possible answers. So one is that in Sherlock, at least, you actually usually can predict the event is about to end. It's like, and this is I've started like noticing these now when I'm watching TV, that they usually give you, there's usually a decent amount of cues during the last 5 seconds that the event is about to end. It's not always true, right? Sometimes they'll purposefully have a car crash you don't expect or something, right? But so there is some anticipation, and I think the bigger issue here too is also just, since this is fMRI data, we don't have super great precision about where these event boundaries are, right? So we only know the event boundary to within sort of plus or minus 3 seconds at best. And so I think a lot of this is just sort of wiggle in the estimation. But yeah, so it's a good question about, right? So it's certainly shifted to the right?
There's more, the statistical test I'm doing here is whether there's more activity after the boundary than before. So it's definitely shifted that the sensitivity is mostly happening after the event boundary. But yeah, the fact that it does is coming up a little bit early is something we got to resolve. Okay, so this is so far only talked about stuff that's happening during perception, right? So I'm arguing that while you're watching this movie, something's happening at these event boundaries that might be this event getting encoded into long-term memory. So now I'm going to ask what happens during free recall. So the other half of Janet's data set is people doing these free recall tasks where we have each individual person giving their description of the movie they just saw. And so again, the way that Janet's analyzed this was looked at the words people were saying and figured out what part of the movie they were talking about and looked to see if their brain patterns looked similar to the movie. So again, we can try to do this in an automated way, except rather than between movie and audio, now we're going between movie and recall. Right? So we have, we break the movie up into sequence of events, so we look to see if the same sequence of events appears when people are trying to do this recall. So one way to plot these results is on this kind of movie recall plot, and Janet showed some of these as well. Right, so the x-axis here is time during recall, the y-axis is time during the movie. And so these little boxes are what Janet showed. This is based on what the words people were saying, what part of the movie are they talking about during each part of the recall. Right, so about 20 minutes into this 20 minutes into this recall, they were talking about stuff that was about 30 minutes into the movie. So what we can do is we can ask the model what time points in the recall do we think are in the same event as the time points during the movie. Right, so this is just using the fMRI data. It never gets access to what people actually said. It just uses brain activity during the movie and brain activity during recall to try to line them up. So here's one of the benefits. They don't always look this good, but some of them do. Right, where we can, so in green here are the time points that it thinks are in the same event with each other. The strength of the green is basically how sure the model is. And so we can find that we track these simulated adaptations pretty well. Meaning that, right, with just by looking at your brain activity during recall, we can predict what part of the movie you're talking about. And so this is a fit into your medial subject and into your medial region. And we find that, again, it's only these long time scale regions where you can actually find this. If you look at auditory cortex, you don't see a significant read statement of this event sequence. Okay, so the last thing we looked at, and this is just kind of a fun analysis, but does, I think, give us some ideas for other global applications of this method. So in this data set, people we have people watched Sherlock and people that didn't watch Sherlock and instead heard audio narrative. And then we had the people that watched it. This is from Asya Zadmood. She had the people who watched Sherlock then listened to the audio description. So now we have two groups of people listening to the same exact stimulus, which is the audio narrative. And half of them have seen the movie before and half of them haven't. And so we can ask, if we run the model to try to line up these two groups of the movie, do we see a difference in their alignment? So what we found is that if you have seen the movie before, there's a slight upward shift on this alignment plot, meaning that when you're hearing the same stimulus, two groups are hearing the exact same words in the stimulus, the people that have seen the movie before are a little bit ahead of the people that haven't seen the movie before. Right? Both of them are both pretty close to the annotations, as they should be. Right? They're both listening to the same stimulus, so they are thinking about the thing they're supposed to. But they are, these events start a little bit earlier in the people that have already seen the movie before. Right? So we're calling this anticipatory reinstatement, although it could be that they're really thinking ahead, or it could just be that as soon as the narrator, audio narrator, starts saying something, they immediately know what they're talking about, right? They know what the rest of the sentence is going to be. So it could just be that they're much faster on the draw than people that haven't heard before, right? Who are trying to construct it for the first time. So this is just one searchlight. So again, we run the searchlight. We see these distributed regions as well as some medial prefrontal cortex, is where we see the biggest shifts on these kinds of alignment plots. Right, so this is the kind of analysis that is really hard to do without a data-driven method, right? Because the stimulus for these two groups is identical. They're both hearing the same audio story. So if you tried to do any annotations on it, it'd be hard to figure out how to make the annotation shift or something, right? So here we're basically letting the data tell us where the shift happens. You can also see too, the shift is kind of uneven throughout the story, so just using like a constant offset is also not a great model of this. So putting all these results together, it's trying to argue for this idea of,
Why do you think the shift is so little?
It's interesting when people's different reactions. Some people say this looks so little, some people tell me this is ridiculously large. It's unbelievably large. That like they think the results are real because it's too large. So the shift here, this is the whole movie on the y-axis. So the shift here is like 30 seconds to a minute or something, right? Which is pretty far ahead in some sense if you're listening to the same story. Right? Most of the coupling we've talked about to these audio stories is like you can align subjects to within like a few seconds or something, right?
So cases where they jump way further in time or not?
I mean, so the average. So the biggest jumps here are relatively large, right? Like if you really, I mean, again, there's estimation errors and stuff in here too, right? But if you really believe like this jump here, that's like you're like 4 minutes ahead or something like the other group. And so, so if that's right, that's very large. I should say here too, this is it's a corrected searchlight map, but these are just random searchlights they pulled out, and so these are almost biased upwards. So it's probably the shift probably not that large. So it's relatively subtle. You can run the analysis a few different ways and still get significant results. So I think it's a stable result, but it's not a massive shift. So all these results I think are pointing towards a view of what's happening during naturalistic perception, where we basically have lots of sort of low-level sensory changes coming into visual auditory cortex. And we start constructing these longer timescale events, right? So I guess it's very compatible with the kind of timescale stuff we've been talking about, but a little bit different because it's really claiming that these events are very discrete, right? And so this is a kind of cartoonish picture that these events are not quite this discrete. But it's basically arguing that it's not just that these higher level regions sort of have slower oscillations, it's that they really have longer periods of stability, and they do still have rapid changes, it's just these rapid changes happen less frequently. And then the memory connection is that it's these regions near the top from these top couple layers that when we hit an event boundary get encoded into long-term memory. And then during reinstatement or during anticipation, we can read these memories back out of the hippocampus.
So do you think that there's like any amount of occurrence that they're like feeding back? Like you're kind of framing this as hierarchical going up, but you think it goes down also?
Yeah, definitely. So I think that's probably one of the reasons for building up these long events is that it helps contextualize the other things that are coming down, right? So I don't have the hours right, but definitely there should be influences of this. For this region, for example, when it gets its input, it should also have textual information coming down, right? So this is a way of getting these lower areas to sort of have a little bit of longer memory, right? So they get to receive information from these longer timescale regions. Interestingly, so we started playing with, there's a neural network model that this is from Yoshi Benju's group that basically does something very similar to this. It takes text and tries to divide it up into these regions. And the way that they operationalize these top-down things, which I think is an interesting one, is that whenever an event starts in some region, it gets to look at the layer above it for how it should initialize itself. Right? So the idea is that when you start a new event, you sort of flush everything out you ask the region above you for the current context, and then you start integrating bottom up after that point. So that's just a random computer science model.
It's a really cool idea for like a deep learning architecture, because before like it's just like the short amount of memory, but then if you're looking at you have these multiple timescales and anything it can learn other new ways of.
Yeah, that's right. So they have like a 3-layer version of this. This is just so it's like 3 layers of LSTMs, and then it's learning. You only pass information up whenever you hit that boundary, right? So the idea is that it makes it a little easier to compute, and also that your gradients don't get messed up because these long timescale regions only ever get information very infrequently, and so like to them it's just like time is really slowed down. They just only see inputs very infrequently, and so it should be easier to learn long timescales. So yeah, they sent to that, which those models are similar to this spring thing. We don't know, but that's one big project we have right now.
That's really cool. And also, like, what would the feature representations be of these like really long timescales too? Like a schema or
Yeah, that's right.
That's really awesome.
So, apropos the last slide, so you showed us in the posterior medial cortex, given that's one of the sort of medium to long timescales, I wonder whether in different regions of the brain you would find different lags corresponding to the time scale of that region.
Right. Yeah, so I mean,
predictive representations, but for my region's specialized time scale.
Right. Yeah, so it's true. It could be that this anticipation is happening everywhere. It's just so small in some regions that we can't see it. Yeah, I don't know. I mean, this requires, in order for this to happen, you need to be, it requires episodic memory, right? And so it could be that you really need to be pretty directly connected to like the hippocampus or medial temporal lobe in order to see this. Like, I don't know exactly how far back those things go.
Back or forward?
Back. Yeah, I guess the other question too is about that. For these lower level regions, so for like the early auditory cortex, for example, is almost certainly like very locked to the auditory properties of the stimulus, and so it's not going to be pushed by this at all, right? And so, yeah, I think there might be an interesting gradient sort of how the separation starts happening. And I mean, you could also wonder about like to what extent you really need like conscious attention to really make this happen, right? It's sort of a construction process, and so. Right, you might, for example, if a person's not paying attention to the narrative, it's very likely this is going to work.
Yeah, I'm more interested in the different scales of predictive.
Oh, I see what you're saying. Yeah. I mean, there's a little bit of that already, so that the anticipation effect is much larger in MPFC than in PMC.
Yeah, I could imagine, because MPFC in general has longer.
Right.
Yeah, I don't know if we can't tell from this whether you should expect a gradual shift across the hierarchy or not. Because I could also imagine, given the timescales you're talking about, like a minute or 4 minutes of prediction, that is people thinking about the next scene because they know what's going to happen next.
Right.
And I don't expect to see anything.
But that's what I mean by predictive representation. So they know what's going to come.
But then you wouldn't expect to see any anticipation in lower level areas.
Depending on what, for instance, if there's a moment where it's going to do something very sudden, it's actually useful because you're going to reduce prediction error when that happens. So if I know that already something like a train is going to pass or the car crash is coming, it's good for me to prepare for it. So I'm pretty sure that especially perhaps for event boundaries, you might see lower level time scale.
But these people are listening. They're not going to see the movie.
Yeah, but still, they can still be predicting what's going to be said, something like
But
that.
it's not at the same level.
And then there was an explosion, and I'm waiting for it to come instead of just.
They know that what they're hearing is not identical to what they saw before. But if
But
they're
they don't
watching a
relate
movie,
it.
pardon?
But they know it's related, it's supposed to.
I'm just saying that your idea of going to the lower regions would probably be better tested in the case where they watch the movie twice, rather than the
I
case
agree.
where they hear a audio narration of an unknown speaker moving at an unknown pace.
For visual predictions, I agree with you. For precise V1 predictions, but we know from a lot of the works in everything from MBPA that you talk to me about a face, I'm going to restate a face. So if I'm expecting that at a point a huge face is going to appear like in the sky or something in a movie, then it makes sense that I'll have a predictive, as soon as somebody says, and then the balloon moves aside, and then I'm expecting for a huge face in the sky.
I will say we have one data point on this that is yet unpublished, which is Miriam's data set, where she has people watching the same videos 6 times. So you don't see, we look exactly for this kind of anticipatory pattern. I'm afraid.
Have you tried?
It's really strong.
Have you tried with this? I guess maybe we wouldn't need the HMM in this case. You don't have to line
Yeah,
that up.
we just looked for like one TR lag.
Yeah.
That would be perfect for this analysis.
Yeah. I mean, yeah. This could be a little better because if the lag variable. Okay. Yeah. Let me take. I'll keep pausing here if there's more questions. The rest of it was just going to be about the details of how the model works. So if there's more science questions, we have to take them.
Would you be also able to like investigate individual differences in memory, for instance, like the number of states, or how fast they transition, or even hippocampal activity to like how long they are recalling, how many events they're recalling, etc.?
Yeah, that's a great question. So the fits to the individual subjects and recalls are sort of necessarily have much bigger error bars on them than to the movies and the whole groups. But yeah, this is something we're thinking about a lot. So we have this new data set of people watching and listening to 16 stories and then trying to recall all 16. And so that gives you a little more variance. In this single movie paradigm, almost every subject is like pretty good at the recall, and so it's a little harder to see. But the 16 story one, definitely some people are better and worse than others. So the question is, are these, can we use the fits of this model, for example, to predict, we're trying to predict a lot of different things. I mean, one basic thing is just like, how much does the person talk about the movie? So for example, if we fail to see this encoding bump, right, so if a person's watching a movie and we don't really see a bump at the end of the scene, can we predict that person's not going to reinstate that scene well? So we know that's definitely true at the group level. So scenes that end with big bumps tend to get recalled better. But it would be great if we could send that to individual people as well.
Maybe you're about to explain this, but I'm curious as to why you think like a voxel in the time series, like 3 voxels, like an actual visual shift in a single voxel. Because you're looking at sort of like pattern similarity, change like deflections and pattern similarity through time, right? So like you could get a big difference in pattern similarity between 2 TRs, I think without any voxel really like shifting, right? So is that
Yeah, that's right. Yeah, so everything, all the illustrations that I've shown so far are just totally fake illustrative data to show the point. But yeah, the idea is we're going to have a lot, big region with like 1,000 voxels or something, maybe more, hundreds of voxels. And yeah, the shift is most likely going to be sort of all voxels will change a little bit or something. So it's going to be correlation across a bunch of voxels.
Just coming back to this issue about the hippocampus and feed forward versus feedback. I wonder whether, like in the rodent literature, there's all this stuff about, for example, place cell remapping. If we had a model of exactly when that remapping would occur, it might help us to figure out, well, I guess my question is, how does this encoding process and or event boundary process relate to that? So is the remapping a consequence of an event boundary detection in the higher order area, or is it actually the remapping that's occurring in hippocampus that then sends this trigger back to the, assuming that's kind of our best physiological correlate of.
Yeah, that's a great question. I'm just talking about this idea of the remapping. I mean, unfortunately, I think that my data is going to be hard for me to give a great answer to this. But I mean, I guess my view for a lot of the stuff is that a lot of these, most of this sort of situation model detection is something that, at least traditionally, we would think of as something that's been consolidated into the cortex. And so the cortex itself, in most of the cases, I would think would be capable of detecting that this event boundary should happen, right? So at least I think of the cortex as the one that's doing this event boundary detection. And I mean, actually, the data sets of hippocampal amnesics watching movies would be a great way to test this to see if their event boundaries are normal or not. And then I'm thinking of the hippocampus more as something that is detecting this change in the cortex in some way, either through an explicit signaling mechanism or it's like monitoring or something. And then detects that now the situation has changed, and then would both try to encode the previous thing as well as bring online any kind of spatial resources we need for the new event. So that's a very feed-forward way when we illustrate where the hippocampus is kind of the end of this long trajectory, right? You take away this arrow. It's the end of this long trajectory, and it's really just sort of keeping track of things and not actually initiating these responses, right? At least this temporal data is not, we don't really have the resolution to say this concretely, but right, it does sort of look like whatever's happening in the hippocampus is after what's happening in the cortex here, right? That there's some extended process of encoding. It's not necessarily true, right? It could be that the hippocampus is starting a process here, and the first thing the hippocampus does is kicks off the boundary in the cortex, and then it does other stuff after that. Or, I don't know. But I think that the possibility that this kind of paradigm is similar to things we can study in rodents, for example, with like remapping, means that maybe we figure this out because we can actually do electrophysiology.
Just to follow up on it, so one of the things that always kind of bothered me was that experiment where they put the rat in the laser world, light world, where they can switch it back and forth between two contexts incredibly quickly. That was troubling to me. And I was like, well, I'm saying that there's this like long time scale thing, and then the hippocampus is near the top, can be super slow, and yet the hippocampus can switch its location like, I don't know, like 5 times a second or something like that. So maybe actually the hippocampus is helped by the fact that precuneus has this also high region and slow time scale. It just never in the world will ever recede. It'll just, the filtering in time, or slowness in time, prevents that from ever happening in the world. It's only with optogenetics or with some kind of bizarre, wait, was that a behavioral experiment? Oh, so I'm like, great, so this is, so then the question is, I guess, like, in humans, could you do this, where you like somehow change where they are incredibly fast, but you somehow get them to actually absorb that? And maybe there's some limit to how fast a human can absorb an event, right? It's like, I cannot change rooms faster than...
Have you been online?
Video games.
Yeah.
That's true, like speed running through a video game, the only thing I know about it...
I don't think it gets up to real... A lot of that's procedural. Those speedrunners, they're not really absorbing the situation change there.
Yeah, it's all integrated probably, but the only thing I know is that there has been some people doing mediotemporal stimulation, and this is one person, one site, so take this as a grain of salt, but that whenever they stimulated its location, he said he felt like he was in an Italian pizzeria and everyone's Italian. And like, so it certainly initiated place change. It was like sort of power gyres through the hippocampus, right? So you can imagine, maybe turning that on and off very quickly, how fast can you teleport in and out of the restaurant.
Just one comment in the experiment you're highlighting, Chris. There was no behavioral readout of whether the animals could sort of distinguish which state their hippocampus was in. So an alternative interpretation could be that rapid switching was part of some sort of inference process, right, where the animal was trying to figure out, like, whoa, what's going on, what context am I actually in? But that itself was not powerful enough to influence what either the animal might do or what goes on in lower down areas.
Also, it was dorsal hippocampus, right?
Yeah.
So the time scale of dorsal
If you did
hippocampus
in
is
ventral,
supposed to
it
be
may not
faster
switch at all. Exactly.
The more ventral in rodents is supposed to be the one with the one.
Maybe President. So I just have a few more. So I think I want to show at least a few more of these slides. So usually I don't really talk about this stuff during this talk, but since this is a Method Z workshop, and also I want to talk a little bit about the internals of the model, because I'm hoping to give some ideas about ways you can make related models to this that also solve a bunch of other problems. Okay. So when we're specifying this kind of probabilistic model, there's basically like 2 pieces we need to talk about. So the first one is we need to have a model for what an event looks like in the brain. And so basically this is sort of the first version of this model we're doing. We did like the stupidest possible thing, which is that an event will have some kind of characteristic pattern of activity. And so this event number k will have some pattern of activity as k. And every time point within that event should have a voxel pattern that looks a lot like M sub k, right? So the probabilistic version of this we have is basically that the log probability of seeing a particular pattern of activity is going to be proportional to Pearson correlation with this latent event pattern, right? So the hypothesis here is basically just that all time points within a event should have a pattern of activity that's highly correlated with this latent event pattern. There's also one other parameter here too, this the sigma, which I'll come back to. But this is basically sort of how the probability falls off with the correlation. So if I see something that's correlated at like 0.5 versus 0.1, how much should that change my estimate of like how good of a match this is? The other thing we need to specify is how events relate to each other in time. And so again, we picked basically the stupidest possible answer, which is we're going to start in event number 1 or event number 0. And then at every time point, we're either going to stay at our current event or we're going to go to the next one. So we can never go back. We can only just either stay in our same state or go forward. And we have to end in the last event. Right? So this basically is sort of what we define as like segmentation, right? You have to start at the beginning, end at the end. And then you're going to have to take exactly sort of k minus 1 steps. If there's k total events, you're going to take k minus 1 steps, and you can pick where they are. And so, yeah, so this transition matrix, again, this is just saying, we have to say what's happening to latent states, and we're just saying it has this very simple single band kind of structure. So if you put these two together, you get a hidden Markov model, which has already shown up here before. Right, but so the idea is that what we get to observe are the things in blue down here, which is the brain states. And the things we don't know are is this event structure, and that's what we want to figure out. And so we need to plug in these two pieces, and we need to say how brain states are related to these latent event structures, right? What an event looks like in the brain, and how these events are related to each other, which is that they're just marching ahead in time. So you can imagine lots of different extensions of this, right? So if you want to allow the transition matrix to be a little bit out of order, for example, if you want to model recall paradigms where there might be some jumps. You could also imagine an observation model that's more complicated than this, that does things like maybe you think that an event shift is going to involve a very small number of voxels, and so you actually want to learn some type of covariance about like which of the voxels in the pattern are important. Right? This just does the correlation over the whole RY. It doesn't try to learn which part of the RY might be informative. You could also imagine a kind of hyperline version of this, where there's some shared pattern of activity that's in this hyperline space, and every subject is going to be a rotation out of that space, right? And so this observation model would also include a rotation matrix of how do I align this subject to the group, right? So there's many. So this is kind of the nice thing about probabilistic models is that it like makes it very obvious what your model is and like which pieces you could switch to generate new models.
Yeah. Is it supposed to be whole brain or just for a particular?
Yeah, that's a good question. So the only ways we've really run this so far is either a searchlight, so that means you do this independently on lots of little regions, or you could parcelate the whole brain into like a relatively small number of parcels, like 200 parcels, and then the voxel activity patterns here would be parcel activities.
Because you have other constraints, right? Because you have higher level events should sort of supervene on lower level.
Yeah, that's right. Yeah, that's right. So the way we did it now is we just like fit the models totally independently and then observe that there was this similarity, but you could force it to find a hierarchy of events, right? So you fit it simultaneously in regions and you say that some events have to be inside others or something. So the last question is how you actually fit this model, right? So the data that we have is the blue stuff, and the things we don't know are this latent event structure, and that's like really the thing that's one of the things we care about. And the other thing is we don't know these M sub k's. We don't know what the events look like. So how can we estimate these two things? So if we know where the events are, then getting the estimated event patterns is really easy, right? So if we know where the boundaries are, we can just average all the time points within a boundary, and that'll give us an estimate of what that event looks like, right? So that estimate is pretty easy. If we had S sub k, we get M sub k pretty easily. If we have M sub k, we can get S sub t pretty easily also. So if we know what the events look like, figuring out where the best place into the boundaries is also not that hard. So it's more complicated than averaging, but there's basically a hidden Markov model procedure you can do that just takes that's relatively quick to compute that just says what's the optimal assignment of each of these time points into event bins if I know what the bits look like. So the way we do this is basically by iterating back and forth between these two things, right? So what we start is we start with an estimate of S and K that like all the events just look the same, and then we try to guess S and T, and we go back and forth until we converge to a solution. And there's one more piece of this that makes the solutions much better, which is this little sigma parameter. And so this basically, this parameter basically says like how tight the events are, right? So if this sigma is very, very high, then it means that we basically ignore the correlation values. So if sigma very high means that all the events kind of look the same. Sigma very low means that even a small drop in correlation means you're not in the event anymore, right? And so what we can do is as we're going back and forth between S of T and S of K, we're going to start with a high value of sigma, meaning when we start this iteration process, we're going to assume we don't know anything, which is true. And then as we start like learning a little bit, we're going to start turning down the sigma. And then once we're like really sure where things are, we can make the sigma very small. And after a certain point, once you start making sigma too small, then the likelihood of the model starts going down, so you can just stop right there. So if you look at like where we're estimating the events, right, what we start out is we don't really know where the events are. We know event 1 is somewhere near the beginning, and event 2 is like probably a little bit after that, and event 5 is near the end or something, but this is just like from the priors over segmentations. And then as we start fitting the model, we start making guesses about these time points and these event patterns. And eventually, this is like a simulated data set where the events are exactly perfect, but we'll start getting more and more confident about where the events are. So none of this is stuff you have to worry about if you want to use, like in the tutorial. All the stuff is inside the package. That it basically has a little schedule where it like turns down the sigma and makes everything pretty.
Yeah. Is the schedule for how sigma changes kind of fixed? So it ends at the same point, or could that, because like another framing of this is instead of fitting like the number of events for each region, you could find the appropriate sigma for, like you could fix the number of events and then find the appropriate sigma.
Yes, that's what we, so the sigma stop, you can tell where the sigma stops once the log likelihood peaks. So it does find, it optimizes the sigma. And you can look at what the sigma is, and it'll tell you a little bit about like how good a fit there is.
It's kind of like how blurry the event boundaries are.
Yeah, that's right. Yeah. That's right. It's how it's right. I mean, so it'll definitely relate to how blurry the event boundaries are, right? It's basically saying like how tight these events are, right? So it's basically saying like how good is this model that all of the time points within this event really look similar to the event. So here's a little animation of what this looks like. So here's a picture. This is just a data matrix. This is a time point correlation matrix. So I'm taking all of the spatial patterns at every point in time. This is just for like a few minutes of movie. Maybe this is, let's say it was 400 time points. So so 10 minutes of movie. And then correlating all those patterns. And so it may be hard to see if you're in the back here, but there's these little blocks of yellow along the diagonal. And we'll talk about this more in the tutorial too. But so these little blocks along the diagonal are the kind of thing this model likes to see. Because a block along the diagonal means that all of these patterns that are close in time to each other all have similar patterns. And then the edge of the block is where the pattern changes. So these little blocks are like the thing that the model is trying to capture. And so I'm going to show a little animation here. Along the top, I'm going to show you the segmentation of where which time points the things are in which events. And on the left, I'm going to show you, so those top things are like S sub Ks, right? What event we're currently in. And the thing on the bottom here is going to be the event patterns. So they're going to start out white, and as they become more certain, they'll get colored in. Right, so what's happening here is we're like going back and forth. We're estimating the thing on the top and the thing on the bottom. And as we're doing that, we're trying to make ourselves more and more certain about one of the answers. So just one reason I show this is to give you a little sense for like how this works, right? Is that it's not like it tries to estimate the events in order from beginning to end, right? What will happen is that it'll basically latch on to some events in the middle that it thinks are really strong, and then it'll try to fill in the other ones, right? And so and you can see even at the final solution, there's some time points that are still kind of white where it doesn't really know what event they're supposed to be in. And
you can also, if you look at the event patterns, you can see that they're relatively diverse, right? So there's certainly some that look like each other, right? But it's not like it's just oscillating back and forth between two events, or between two patterns, right? The events are in a relatively high dimensional space. Yeah.
I don't want to derail from like you're going, so you can just save later. But so we've been looking at all these plots as well in our lab, and one thing that I think is just the event segments are really obvious and really beautiful on this plot. But there's also on the off diagonal. It's basically, it looks like it's the same pattern again. And do you have any thoughts about what that means?
Yeah, that's right. So definitely, yeah. So there's like this model basically only ever looks at structure that's like really close to the diagonal, and this stuff never even enters into the computation. But yeah, it certainly looks like there's interesting stuff out here, right? So like this little block here means that these two events that are pretty far apart in time had similar patterns. So there's a lot of possible reasons. So I mean, one thing is that the fMRI patterns are, of course, just a projection of like the real pattern, and so it could be a sort of coincidence these two things have the same fMRI patterns, but they don't actually have the same underlying neural states. The more interesting one is that those regions actually do depict similar situations in some sense, right? So we could try to predict which events have similar patterns based on some property of the movie. And so we've done that to a mild amount of success in the sense that you can predict at least above chance amount of the variance with like sort of simple predictors about like, are you inside or outside, or like, is Sherlock on the screen, and those kinds of things. But yeah, we don't really have a great, like a super predictive model of that yet.
It's related to the figure that you showed earlier, the animation with the 9 spatial modes. There's a little
Yeah. Yeah, that's right. That's right.
The way I've been interpreting the off-diagonal stuff in my lab is like it's like memory loops. So you can think of revisiting a past state.
Mm-hmm. Yeah, that's right. So I mean, it's a question whether it's like a bottom-up or a top-down thing too. About right, is it that the input is similar, or you're like remembering something that happened before? Yeah.
There's also something confusing about the off-diagonal dark blue spots.
Mm.
So that's like this point is really different from that prior point in your past. It's like a repelling thing.
That gets really methodologically crazy.
Yeah, that's whether those negatives are meaningful is a bit of an issue.
There are some stimuli which might tease that out better than others,
Mm-hmm.
which is dependent on what the sense of temporality is within that stimulus. So in a movie, the narrative, each point in the narrative depends on the previous points. But in something like a piece of music, like a complicated piece of music, where you might have a theme that's then reinstated in a different instrument, in a different context, you're sort of moving in two directions at time at once or something like that. You could design a stimulus that you would expect certain off-diagonal properties.
Yeah, that's right.
Maybe one other relevant thing here is like you can do this, you can do RSA between the movie and the recall data, and it works pretty much throughout the recall network. So there is at least some semantic content that's responsible for these.
Right. Yeah.
I was wondering if you looked at things like emotional arousal or like fear responses or something, because those things could also provide
Yeah.
overarching context that might make.
Yeah, so I've basically looked at the regressors these are like the ones that Genesis RA Esther made, and so there were like one of them is like emotional valence. It wasn't a huge predictor for this is just like one region. This is a PMC region, but I can imagine it would be for other regions that care more about it maybe. But yeah, I mean it's a little bit of a fishing expedition because we don't know exactly what the right things are. And some of these things also like valence have like a pretty big impact on univariate responses, but maybe right that these like emotional areas really come online if there's a lot of emotional valence, but maybe there's not much represented in the patterns. And so yeah, we really don't have a model that works super well yet. Yeah.
You put some constraint in the condition metric that one state cannot go back to
Mhm.
because of the correlation is
No, so you can do a version of this where you're allowed to revisit states. So the main reason for doing that is so that we could align different data sets, right? So we have the movie and the audio narration, right, or movie and recall. And so if we force both of them to go through the events in the same sequence, then it makes the alignment process much easier. Right, if you allow jumps, then any kind of off-diagonal thing like this is going to really mess you up because it's not going to know, right? If you try to align two data sets and this event comes up, it's not going to know like what the correspondence is, right? So in some way, the limitation is just like the simplest version of this model to make it so that you can align across data sets. But certainly the recalls are not perfectly in order, and so this model fails to capture those pieces of it. That's right. That's right. Yeah. Mm-hmm. Yeah, that's right. Yeah, so you can basically do a cross-correlation matrix of these, and you do get, right, it's basically just, you get the same pattern that you see here. It's just with these boxes are now one box, right? These little rectangles are now one box. And yeah, then you could try to ask, like, which scene, can we predict which of these scenes are going to be similar? And the answer is a little bit, but yeah, that's right. Did you have something? Yeah. Mm-hmm. Yeah.
So just by eyeballing this and not having done any clustering algorithm on it, it seems like there is a bunch of things that are, so there's something at time point, I don't know, 325 and something at time point, I don't know, 125. And those things keep like, there's two.
Yeah.
So I wonder whether.
Yeah, so if you look at the scene, yeah, definitely there is some.
If they said
Yeah.
whatever was happening in the movie or the narrative, once based on the data, the clusters are identified, one can see what was going on at these time points specifically.
Yep. Yeah, that's right. Yeah, so that's exactly what we're trying to do with these models. We basically just don't, our current annotations of the data don't do a great job of predicting this similarity, and so it's a lot to know. I mean, there are also, there's a little bit of a method issue here too to think about, is that this model is going to prefer to find events that are like really dissimilar from each other, because if there's two events that happen right next to each other and they're kind of similar, it's going to lump them into one latent state most likely, right? And so it's a little bit incentivized to find bigger jumps, and so the events it's going to find are going to tend to be relatively dissimilar. But that's not necessarily a bad thing. It just means that, right, the kind of stuff we're going to try to predict on this are going to be like big changes rather than small ones probably. Yeah, I'll do one more question. Mm-hmm. That's right. So yeah, so the transition matrix is fixed. Yeah, so that doesn't get estimated. So yeah, what's getting estimated is the main thing that's getting estimated is basically the underlying event vectors, right? What is these things, and we're also estimating this, like which vectors go into which time points, right? So the transition matrix is fixed, but we don't know when the events switch, right? So these two things are the two things that are getting estimated. Well, that's a great question. So yeah, so how do we get the number? Everything I've told you so far doesn't answer how do we actually know how many events there are, because that's something we want to know, right? So so there's a bunch of ways you could do this. The super fancy way would be to put some like non-parametric prior or something on this, and I did that in my last project and hated it for like 2 years. So instead, I tried to do something easier, which is we're just going to try to look for boundaries that correspond across subjects. So what we're going to do is take all but one of the subjects and learn boundaries on them. And then we're going to look in our last subject and ask, do the boundaries show up in this last subject as well? So you have to have some measure of do the boundaries show up, and the simplest thing we can think of is just looking at windowed correlations. So do we see a big change in pattern correlation at these boundaries, right? So we're going to define the boundaries based on all but one of the subjects and look to see if there's a big pattern shift in the held out subject at that time point. So what we can do is just build a whole bunch of models at different numbers of k. These are all just independent models. And what we find is that, for most regions, this is a plot from like posterior medial cortex. I forget. Yeah, or angular gyrus maybe. I forget which one. This is probably angular gyrus. It's a little longer. And so, right, so we can see. So first of all, you see all these values are very far above 0. So in almost all cases, like the boundaries that we predict most of them do show up. But there's a peak for most of these plots, right? So meaning that there's a sort of optimal number of events, and we can't say exactly, right? But it's like sort of for this region, it's like in the 70-ish range, right? Is the best number of events. If you do too few or you do too many, then the boundaries don't correspond as well to the held out subject, right? So we're just doing kind of cross validation on this event parameter k to figure out how many events there are. That's right. So that's the way that we get this this very first map an hour ago. So the way we get this one is, yeah, this is showing the best fitting. Where's the peak of that curve for each searchlight? And we see that it shifts, to the left as we go up the hierarchy. Yeah.
I'm always one slide behind. I'm wondering whether, so the off-diagonal stuff that you showed us, all of this is in one sort of brain region. I wonder whether there are off-diagonal things that are so that appear in multiple regions, so they have a higher reliability than just, oh, the pattern happened to sort of.
Yeah.
So if PFC and sort of posterior medial, and I know a bunch of them are saying this thing again, maybe it's more reliable that there's actually something like time travel that Jeremy
Yeah.
was kind of alluding to, whether predictive or post-predictive.
Yeah.
As opposed to.
Yeah, that's a great question. I mean, there's a few ways you could do that. One way we've done a little bit, which is like what Kieran's done in his stuff, is you just take like a whole bunch of ROIs and just concatenate them together into like a mega ROI. And so in that ROI, that mega ROI, if you see something that's very similar, it means it's similar in all the regions at the same time. So I mean, yeah. So in some ways, it's just a way of getting to a higher effective dimensionality, right? So that you don't have as many collisions, or the collisions that you do have should be meaningful. And so yeah. So definitely, sometimes we like to be sort of splitters and look at a little tiny region of the brain, but like if we really care about is the brain cognitively in the same state, we really need to look at bigger networks.
It would be basically like a caption for what the PFC is talking about, what the posterior medial is talking about, and sometimes they're talking about the same thing.
Yeah, that's right. Yeah.
Sort of mentioned the next question before, but I don't know if you're finding, I think you mentioned you've done it, but letting like certain groups of subjects have different Ks and similar Ks now, is that something about seeing partial recall or amount of
Yeah, that's a good question. Yeah, so for the movie stuff, usually just assume that everyone is identical during the movie, which, we know is probably not a terrible answer thing, but is definitely not totally right. I mean, so even if you just behaviorally, if we ask people to divide the movie up, different people will divide it at different levels of granularity, right? And you can direct them, but just people's like default level seems like kind of different across people. And it could just be some people are lazier than others when they do the event boundaries, or it could be that people actually have some difference. Like some people really rely on their, so it could be that their timescale areas are a little bit different, or it could just be that when people do this tasks, some people use their angular gyrus, which is like a 40-second thing, or some people use PMC, which is like a minute and a half or something, right? So it could be that people are very flexible about which region they're reading out of, and so the Ks could be the same even if they're like using them differently or something. But yeah, it's a good question. Yeah. It gets a little bit tricky to compare. I'd say in general, so if you use like this metric of like how well we're doing out of like a held out subject prediction, there's not a huge difference. So I've definitely looked at this across like the couple little ROIs I put down on the surface, and they all have relatively similar held out correlation values that are in the same range. Yeah, I should say like if you look at like hippocampus, for example, we're not able to find good things. It's like way down at 0. So there's basically some regions that we really can't fit, and a lot of the other ones have sort of similar things. It's hard to say. I mean, in some sense, visual cortex is better because there's lots of boundaries, and it's able to lots of boundaries go across subjects. Whereas these ones, the boundaries that we have are good, but there's not as many of them, right? So it depends. There's more good boundaries in the early sensory cortex than here, but it's clear if that's maybe just like biological fact. I don't know, but. So yeah, just to sum up, right? The model here is this hidden Markov model, where we're trying to infer both when events happen and what they look like, and then we can just do this cross-validation hack to figure out how many events there are. And so yeah, so all the inference stuff is already in the package I'll use in the tutorial, so you don't really have to worry about that. But I think there's lots of ways you could extend this. Almost all this stuff has already been brought up by people, but right, to understand the relationship between different events within a story or across stories. And then I think there's lots of other like potentially crazier extensions of this, and so I'm trying some of these now. But right, the model really is not that fMRI specific. The only fMRI specific part of it, I mean, even this is not that fMRI specific, but is this, right, what the brain looks like in relation to the event. But you could imagine that instead of fMRI here, you have another neuroimaging modality, or you have something that's not neuroimaging at all. So we have like what the word vectors are that the person's saying, for example. Or maybe simultaneously we have both measures of behavior and brain states, and we could put all of those into the Markov model. And then if we want to have even fancier observation models, we could do things like hyperlon across subjects and things like that. I just wanted to end with just a brief plug. I'm currently setting up a lab at Columbia, looking for people that are interested in doing this kind of stuff. If you or someone you know might be interested, definitely let me know. I haven't really started seriously recruiting yet, so you can get in under the wire of I'm not currently drowning in stuff yet, so please let me know. Thanks.